Hello, college biology students. Hopefully you watched the other video first that I posted on the electron transport chain. It goes into a lot more detail than we need, but it does a really nice job of explaining how that energy from electrons is transferred through this sort of chain of events. And it talks about this 4-protein complex by which that happens. So this is located in that inner mitochondrial membrane, which, remember, when we looked at our mitochondria, we talked about that structure a lot. Oh, that's a terrible mitochondria. Oh, let's just pretend it's fine. This inner membrane here with all those folds, we said this is called the cristae, because all those little folds are called the cristae, and this is to increase that surface area. So the whole point of this is to make ATP. The whole point of this is to make ATP. So here's the 4-protein complex that participates or functions as the electron transport chain. And remember, in the video they talked about how these 4 proteins are repeated tons and tons and tons of times throughout this inner membrane. So there's 4, 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3. And remember, this is just a 2-dimensional drawing, a 3-dimensional membrane. So we have many of these 4-protein complexes. And it talked about something called chemoosmosis, which we'll define in just a moment. This, by the way, is an enzyme called ATP synthase. It's an enzyme. Enzymes usually end in ASE, and they're named for kind of their job. So ATP synthase is an ATP synthesizer or maker. So this is where the ATP is made. I love this diagram because you can see it talks a lot about all of these hydrogen ions, which are literally just naked protons. Remember that the hydrogen atom itself is just a proton and 1 proton and 1 electron. That's all hydrogen is. So if hydrogen loses its electron, it's a hydrogen ion. It's literally just a single proton with no neutrons, nothing else attached. So what we see here is we see the concentration of hydrogen ions that are then being pumped through this membrane. The other thing that's really important are all of these carrier molecules. So you can see, so this NADH, it's all about the NADs and the FADs, really. So the NADs and the FADs. So NADH was built by taking a NAD plus, adding a couple of electrons and another proton, which is the hydrogen. So as this transfers its a couple of electrons, boom, boom, to this complex. So now we've provided some energy here by giving these 2 electrons to this first protein. And you can, I don't know if you can see this line, this indicates that this energy kind of flows all the way through here. So NADH gives up a couple of electrons. FADH2 also gives up a couple of electrons and becomes FAD plus. And so we see these electrons powering this process, moving through this protein complex in the inner mitochondrial membrane. And these electrons are powering the pumping of these hydrogen ions or protons across this membrane from the mitochondrial matrix to the intermembrane space. And as this happens, it's creating this gradient. Remember how much nature does not like a gradient? It's creating this gradient of all of these protons on this side of the membrane and not so many on this side of the membrane, right? So we have a lot more of the protons, a buildup of charges that's happening in this inner membrane space. Nature doesn't like that concentration gradient, and it's sort of like, I always like to compare it to a lightning storm. When you're sitting outside in the summertime and things are brewing and you start to see the way that lightning actually happens is you have a cloud over here that's, you know, really charged, and you have other areas in the sky that are the opposite charge. Nature doesn't like that. And this is a very, very simplified, it's a little more complicated than this, but really lightning happens because, boom, we need to get some of the protons, this buildup of protons, equalized out. So we probably, protons need to go this way, and the electrons needed to come this way until, and that lightning occurs to try to equal that out. That is exactly what is happening inside your cells. It's miraculous that if we can understand how lightning works, we can understand how ATP is made. This buildup, this concentration gradient of positive ions, then moves through this fancy enzyme called ATP synthase. And when it does this, it's like a little battery generator that takes this adenosine diphosphate with two phosphates on, plunks and causes another phosphate to attach, and it becomes ATP, that energy molecule for the cell. So it is all about, you know, electrons, some carrier molecules that are transferring electron energy to these complexes that then use that energy to pump protons through the membrane, which make this concentration gradient. This is a problem for nature. We don't like this. So these are used almost as like little, it's like an opposite charges on a battery used as a little, as an energy to convert that ADP to ATP. So it phosphorylates that ATP. We talked about phosphorylation earlier. So let's go through this process just really quickly. So we talked about chemoosmosis, that movement of ions through a membrane due to a concentration gradient. So chemoosmosis literally is the concentration gradient. So you have, well, it's movement through because of that concentration gradient. So we talked about our mitochondria. We know I'm not an artist, so suffer through this. This is my mitochondria. This is the inner membrane that has all those folds. And we end up with, because of that 4 protein complex that are in here, we end up having a ion concentration buildup here, which we said nature doesn't like that. And then we have this beautiful ATP synthase. It's fancier than that, but it is powered to make ADP from ATP, or it make ATP from ADP by, it's powered by moving these ions through, acts like a little battery pack. These proteins are actually doing the opposite thing. They're taking positive ions, pushing them out here, so to cause this buildup. And they're powered by those NADH and FADH2 molecules. So the first step, there has to be this chemoosmosis, so we have to understand that. And then the first step is that NADH and FADH2 donate their electrons to these 4 carrier molecules, and these are located on that inner mitochondrial membrane, as is represented by beautiful little pink circles. The step 2, these pump those hydrogen ions to cause this buildup of concentration between the inner and outer membrane of the, remember, this is the mitochondria. Oh, sorry for my It is not beautiful. I know you all suffer because of it. And then this causes that buildup, number 3, and that buildup causes powers like almost like a battery, the ATP synthase to make ATP from ADP. And then the other thing we want to remember, that very last step, oh, forgot a period, the last step here, the last molecule in that electron transport chain actually donates its electrons to oxygen, which also accepts some hydrogen ions, forming water. And where did that oxygen come from? You had to breathe in oxygen, and that is the whole purpose of you breathing in oxygen. It is not because it feels good and you like oxygen and you need it for your lungs. Your lungs wouldn't need oxygen if your cells didn't need oxygen. I'm in the way no matter where I go here. So the whole point of breathing in oxygen is for this process right here. So that's kind of it in a nutshell. So if everything goes absolutely right and everything is perfect, we get a lot of ATP from aerobic cellular respiration. We get about 38 ATP. 2 of them are used up in the process because they got to power, we have to use some energy kind of in that process. So we say a net production of 36. That's pretty good. That's a lot of ATP. That's a lot of ATP. So that's kind of it in a nutshell. And then we'll talk about this again, but this process of respiration goes kind of hand in hand with the process of photosynthesis, which we'll talk about in our next video.